All right. Hello, lab. How are you doing today? All right. How are you doing today? All right. We are going to see order of draw today. So, order of draw is the most crucial step in a pre-analytical stage of a clinical laboratory process. So, we want to get this topic today. And order of draw is a technique that is employed in a pre-analytical stage of laboratory process, which entails choosing a proper test tube for the required test requests. So those test tubes are color coded, and we are going to see what that color indicates in each test tubes, and also we are going to see the additives that are in each test tubes and what those additives are going to do in that tube when we put blood in them. So, and also the tubes could be glass or plastic, and plastic is getting more preferable because when we see glass, and glass tends to break, and that break may cause injury as well as infection from blood-borne pathogens. So we want to make sure prevent ourselves, and using plastic test tubes has recently been very applied. And there are different additives in each test tube, and the test tube, some test tubes contain just a clot activator, some test tubes contain anticoagulants, some tests contain anticoagulants and glycolytic agents, and also some test tubes are just plain, nothing in them. So just it helps us to naturally clot the blood and get the required serum out of it by centrifuging. So waiting or keeping that blood for 30 minutes will clot the blood, and after that, when we centrifuge that blood, we get a serum out of that test tube. So uh, so uh, so some facilities have their own preference over the other. So we are going to follow those facilities when we do our job in real place, in real time. And the reason we follow this order of draw is because in each additives there are in each test tubes there are specific additives, and those additives may cause a significant impact on the other test tube. We want to make sure that impact will be minimized by following this order of draw. So there is a carryover that goes from one tube to another. So we want to make sure that carryover is not going to impact the next test results. So we are going to see general order of draw today, and after we draw our blood, we are going to invert gently so that the blood will be mixed with additives in the tubes. So for anticoagulant tubes, about 8 times, and for non-anticoagulant or non-additive tubes that contain nothing, just 5 times. Inverting it gently 5 times will help the blood to be homogeneously mixed. And also we want to make sure to get the required amount of volumes for each test tubes required. So short volumes are not always accepted, especially with a saturated tube, which is a PTT tube, which is an INR testing. So we want to make sure get that 9-to-1 ratio volume of blood, 9 blood to 1 volume ratio of blood. So it needs to be full to there is a mark on each test tubes. We want to get the required amount of blood to that mark. That means it's okay, it's full. And as we go into order of draw, we are going to see the steps that are needed to do our job. So number one is cultures. So culture is number one. So like bacterial cultures. So when there is a bacterial culture on the test request, you wanna do that first of all the test results. So after getting that bacterial culture, you are going for the next test requests. So some cultures are in a bottle, and some use a light yellow SPS tube. A light yellow SPS tube should be light yellow SPS tube. So if it is a bottle, that's very good. After drawing a blood in that bottle, you just send that to the lab, and they will isolate the bacteria, incubate, and determine what's bacteria in that bottle. But if it is a space tube, and that a space tube, a yellow space tube, and you will send that to a space to the lab, and the lab will put that blood into bottles, so it will be transferred for incubation. So we do this first because of sterility that is going along. So it needs to be sterile. It needs very meticulously done. So for this reason, we want to do it first. Sterility is the most important thing when doing bacteria, because you don't want to get contamination from the outside. So due to sterility, it needs to be done first, and it may be required from each arms. So this depends with the facility. We are going to follow our facility's procedure. All right, so number two is going to be a sodium citrate tube, which is a light blue tube. A sodium citrate, a light blue tube, which is for a prothrombin time or partial thromboplastin time. So it's an INR for PT and PTT, which contains sodium citrate. So we want to get that first if there is no blood culture, because there is a hemoconcentration. That is when we put a tourniquet on the arm of a person going to be drawn. There is a hemoconcentration. That hemoconcentration will alter the test results that is going to be done from that tube. So we want to get first that when there is no blood culture. But if there is a blood culture, you can get after blood culture. But if the tourniquet stays on the arm for long, you wanna use a different arm and a different place. And also it's very advisable most of the time to get INR sodium citrate tube without a tourniquet. But if that's not possible, you wanna get that test sodium citrate tube first. And number three is a serum tube. Serum tubes, they don't have nothing in them, no additives. So for that reason, they need to be number two, because of homoconcentration, and that reason we are going to do sodium citrate first, but serum tubes are the next ones. So serum tubes are red, orange, golden, and tiger tabs. So you are going to use red tubes for rare metal determination, cold agglutinins determinations, and you are going to use the golden tiger tab for general chemistry, for thyroid testing, for BMP, and all those general chemistry tests. There is no additives. Some may contain clot activators, but sometimes it's plain, no additives inside. And number four is plasma tube. So these plasma tubes are lithium, they have lithium or sodium heparin enzyme. Lithium or sodium heparin enzyme. So this is required for STAT BMP or STAT electrolytes. So we want to get this number 4, and it contains a fibrin because there is no natural coagulation is going, because we have added the anticoagulant. So the anticoagulant keeps the fibrin, but the fibrins are absent during the in the serum tubes, because the fibrins are utilized during the clotting process. So this has got fibrin in them, and sodium or lithium heparin, and it's a green or light green. The tube is green or light green, a plasma tube, and it's light green or green. And number 5 is a purple or lavender. So this purple or lavender is, there is EDTA additive, which is an anticoagulant, and it's a K2 EDTA in a plastic and K3 EDTA in a glass. So K2 EDTA in plastics is mostly utilized in a lab setting. So purple or lavender is used for CBC, ESR, and hemoglobin A1C testing. CBC, ESR, hemoglobin A1C testing. And number 6 is a white or pearl color tube, which is a K2 EDTA. This test is required for molecular diagnostic testing. White pearl K2 EDTA. This is a plasma preparation for molecular diagnostic testing, like paternity testing, HLA testing, utilized for molecular diagnostics. And number 7 is a gray tube. In a gray tube, you are going to have 2 additives. Number 1 is potassium oxalate, and number 2 is sodium fluoride. So potassium oxalate is an anticoagulant, and sodium fluoride is anti-glycolytic agent. This gray tube is used for glucose determination, glucose tolerance determination. So that sodium fluoride is going to break or prevent from glycolysis taking place. So glucose will be utilized, so it's still in the cell, so we want to make sure that glucose stays intact before we take our test. So the sodium fluoride is going to help the anti-glycolytic process, and potassium oxalate is an anticoagulant. So gray tap tube is for glucose determination testing, mostly for glucose tolerance test, which is a GTT. Thank you very much. Bye-bye.